Hello everyone, and welcome to the 7th student. Today we're looking at the paper 1 from May 2024, specifically time zone 1. We'll be looking at time zone 2 in the next video. So let's get started. So question 1 is saying the image shows the nucleus of a cell from the pancreas that is surrounded by endoplasmic reticulum. What describes this nucleus? Okay, so what I'm going to do in this video is you can pause now, think about the question, but I'm just going to count down from 3 and display what the answer is. So 3, 2, and 1. The answer is D. Why? Okay, so it is composed of highly folded internal membranes and a liquid matrix. That's not true. So a nucleus does not have highly folded internal membranes. They're flat. It contains ribosomes. That's not true. The cytoplasm contains ribosomes, but the nucleus does not. And it also doesn't contain organelles. Again, the cell does, but the nucleus does not. However, it does contain chromatin, that's DNA, and it is surrounded by a double membrane, which is not highly folded though. Okay, hopefully that makes sense. So that's how we're going to do every question. I'm just going to explain why the others aren't correct, and hopefully that'll help you discard incorrect ones in future questions. Okay, so for question 2, the image shows a single paramecium with food vacuoles that contain ingested cells of the unicellular green alga chlorella. What can be deduced about paramecium? Again, 3, 2, and 1. Okay, so it's C. Why? From the information they're giving us, we don't know whether it's a prokaryote. We don't know that it can't perform all of the functions of life. We don't know that whether it's an autotroph or not. The only thing we know is that it's eating another organism. Therefore, it carries out heterotrophic nutrition. So take things at face value with these questions in IB, okay? They can be tricky, but you just need to read every word. And what's not in the question, you don't have to use your imagination, right? Just stick to what's in the actual question. Okay. So question 3, what distinguishes simple diffusion from facilitated diffusion? And again, I'll give you 3 seconds. So 3, 2, and 1. Great. I know the answers are really long, so maybe that took you some time to think through, but remember, facilitated diffusion uses proteins in the membrane, so the rate of it depends on the number of proteins in the membrane, which makes sense, whereas simple diffusion happens across the membrane, so it does not. Now, why are the rest incorrect? Well, A, facilitated diffusion can happen in prokaryotes, not necessarily only in eukaryotes. Second of all, energy is also not required by facilitated diffusion, right? Both are passive processes. And then also, here it says indeed that simple diffusion can only move substances along the concentration gradient, while facilitated diffusion can move along or against. That's not true. They're both along against the concentration gradient. Along the concentration gradient. So that's why it's C. Great. So now in terms of 4, this one should be easy. How is a prokaryote different from a eukaryotic cell? 3, 2, and 1. Great. So eukaryote cells have organelles, therefore they're compartmentalized. They also have a nucleus, whereas prokaryotes do not, and therefore are not. B is false because both have ribosomes. C is false because both have DNA. And D is false because some eukaryotic cells, like plant cells, can have DNA as well. Okay, so now on to the next question. This is question 5. Sorry, this is a mistake. Which molecule can have cis and trans isomers? Again, 3, 2, and 1. It's C. Why? Because remember, cis and trans isomers refers to the double bonds in carbon. None of these other molecules have double bonds between a carbon and a carbon. Therefore, this is the only one that can have cis and trans isomers. In this case, because they're on the same side, it's a cis, right? But if they were on opposite sides, it would be a trans isomer. Okay, moving on to question 6. The diagram shows the amino acids, that's a mistake, present in a molecule of insulin using 3-letter abbreviations. What describes the structure of insulin? Okay, again, stick to what the diagram shows, not to what you know about insulin, okay? 3, 2, and 1. C. Why? Well, it clearly shows that there's 3 disulfide bridges, right? That's part of the tertiary structure. You should know that. And then it has 2 different polypeptide chains. So this one over here and this one over here. And remember, when a protein has more than 1 polypeptide chain, it has a quaternary structure. Now, here it says insulin is a fibrous protein since the amino acids are arranged in a linear pattern. Well, it is a fibrous protein, but we can't know that from this diagram alone, right? Then B, insulin consists of a single continuous polypeptide chain. That's not true. There's 2 polypeptide chains. And D, there is no evidence of a 3D shape in the diagram, so it has primary and secondary structure only. Just because the diagram doesn't show it doesn't mean it doesn't happen, right? So I think that's quite straightforward. Again, if you have any questions about any of these things, let me know in the comments and I'll do my best to resolve. Question 7. So the graph shows the effect of limiting factors on the rate of photosynthesis. What can be concluded from the graph? Again, from the graph only. So 3, 2, and 1. B. Why? Because it shows, so here we have a low light intensity, right? So at 0.1% and a low light intensity is the only limiting factor. That is true. Why? Because when you increase the light intensity, photosynthesis automatically improves, right? Without changing anything else, showing that light intensity is indeed the only factor which is limiting that reaction, right? However, when you look at C, so a CO2 concentration of 0.003 and a low light intensity, both CO2 concentrations and temperature are limiting factors. We don't know that. There's no information given on temperature, so we have no idea. At a CO2 concentration above 0.1, there are no limiting factors. That's not true. It's still the same limiting factors. And then A, at a CO2 concentration of 0.1 and a low light intensity, temperature is the only limiting factor. Again, no, it's light intensity, because when we increase it, we see that the photosynthesis rate improves. Hopefully that's clear. You need to understand what a limiting factor is, right? It's a factor which is the one that's kind of setting the reaction behind. So if you increase it, the reaction increases as well. Okay, question 8. How do human muscle cells respond to intense exercise? 3, 2, and 1. They produce lactate. Yes, so basically what they do is they do the anaerobic pathway, and that involves producing lactate. Ethanol and carbon dioxide are also part of the anaerobic pathway, but not for humans, right? And it's human muscle. They produce smaller amounts of ATP. That's not true. They do the opposite, right? And then they stop using oxygen and replace glycolysis with lactic fermentation. So they do stop using oxygen, but they don't replace glycolysis with lactic fermentation, because glycolysis happens before lactic fermentation. So for lactic fermentation to happen, glycolysis must have happened beforehand. Okay, great. So now question 9. Watson and Crick worked out the 3D structure of DNA. What did the model they built show for the first time? Again, 3, 2, and 1. Okay, the thing is, this is tricky because you need to know what Watson and Crick showed, right? A lot of these are true. So DNA is a polynucleotide. That's true. DNA contains equal quantities of adenine and thymine. That's true. DNA molecules have a helical shape. That's true. But what they showed is that it's a double helix with anti-parallel backbones. So that's just a fact, right? So just make sure to memorize and write it down so you don't forget next time. So question 10, the image shows details of a cell from a male locust, Schizocerca gregaria. What can be deduced from the image? Okay, and 3, 2, and 1. So it contains chiasmata. You can clearly see that, right? Because there's different chromosomes being intertwined, and there's points at which you can see they're connecting and most likely exchanging DNA, right? There's no evidence of circular chromosomes. These are clearly linear. And then in terms of what stage of meiosis or mitosis they're in, if it's chiasmata, right, if they're crossing over, it's prophase 1 of meiosis. So it's neither mitosis nor prophase 2. Okay. Question 11. So Huntington's disease is a neurodegenerative genetic disorder caused by a dominant allele. What can be concluded from this pedigree chart? These are really tricky, so really think about this one. 3, 2, and 1. So what I recommend for exercises on pedigrees is trying to see if the opposite of the affirmation could be true. And if it is, then you know the answer is not correct. So for A, Huntington's disease is sex-linked since most affected individuals are male. Okay, well, is it possible that it wouldn't be sex-linked even if most affected individuals are male? Yes, right? So most individuals being male, the affected ones, is a sign, but not a clear indication that it's sex-linked. So A is not correct. Then B, it's not sex-linked since IV1 and IV3 are not affected. Okay, could it be sex-linked if IV1 and IV3 are not affected? So let's take a look. So if it were sex-linked, then this affected female would have at least one big X, right? And then the unaffected male would have a little X and a Y. Therefore, these two boys, IV1 and IV3, could have inherited the small X from their mother, because she could have a small X, and the Y from their father. Therefore, it doesn't prove that it's not sex-linked. Do you see what I mean? I hope this makes sense. Okay, then D is complete nonsense, because IV2 is a carrier since individual 3-2 is affected. Okay, so if we look at 3-2 here, the mother, IV2 being a carrier, just doesn't make any sense because the IV2 is not even related to 3-2, right? And then it's C, because, so is it possible that it's sex-linked if individual 3 is affected? So if we look here, in this case, it's a small x, right? And then, a big X, the dad is affected, and then the mother is 2 small x's, so it would not be possible if it's sex-linked for the son to be affected. Therefore, it must not be sex-linked. Okay, so I hope that makes sense. I know this is quite tricky. Moving on. So question 12 says, how are target genes transferred to a bacterial cell? Again, 3, 2, and 1. It's D. So this is just factual, right? You need to know that. The way target genes are transferred is they're cut with a restriction endonuclease that cuts at specific base sites, right? And then they're attached to a plasmid using ligase. And it's important that both are cut with the same restriction endonuclease. So the plasmid, you're going to put the gene in and the gene itself so that they'll have the same sticky ends, right? The bases are cut at the same point and then they'll stick together with the ligase. So here B is incorrect because you don't use gel electrophoresis to transfer it. Then A is false because Taq polymerase is not used to cut a target gene, right? It's restriction endonucleases. Taq polymerase is used for PCR. And then C, a nucleus containing the target gene is cloned and transferred into the bacterial cell. That's also not how it works. You want to transfer the gene, not an entire nucleus. So it's D. Great. Okay, one where you have to think a little. So, sickle cell anemia is a genetic disease caused by a single base substitution mutation. 2 parents, both heterozygous, have 1 unaffected child and 1 child with a mild form of the disease. What are the chances that their next child will be unaffected? Okay, again, think a little. And 3, 2, and 1. Okay, it's 25%. Why? Because if the parents are heterozygous, that means they're both big A, small a, you could call it that, right? Then the chance that the next child will be unaffected, that is half little a, is 25%, right? If you do a Punnett square, you'll soon figure out why, right? So the possible combinations are big A, small a, that's 50%. Big A is 25%, and small a is 25%. So, and the only chance that they're completely unaffected is the small a, right? Okay, so moving on. 14, which gas is the main contributor to the greenhouse effect? And this is a tricky one. So, again, 3, 2, and 1. It's water vapor, okay? Most of you might have said ozone or nitrogen oxide, methane as well. So basically, it's actually water vapor, but it's not brought up as much because water vapor is a natural gas that goes into the atmosphere. There's just a lot of it, and that's why it's the main contributor. But it's not because of humans, right? However, methane and nitrogen oxide are because of humans, and then the ozone is referring to the ozone layer, not to the greenhouse effect. So, great, let's move on. In a stable natural ecosystem, how is the supply of nutrients and energy maintained? And this is straightforward, I think. 3, 2, and 1. So it's C. So nutrients are efficiently cycled. So the carbon cycle, right, the nitrogen cycle, and there's a constant flow of energy. So from the sun to autotrophs to then the consumers, and then again from the sun. So it's not recycled in this case, right? It's a constant flow. So B is false. The energy from the sun isn't cycled. It's actually lost in the end, right, in the final consumer. Again, same. A is false because energy is not cycled. And then D, energy is not cycled. So energy is lost, right? It's an open chain. So it starts from the sun and it ends with the last consumer and then it's lost eventually. That's why we need the sun. Like ecosystems need the sun. Okay. So then question 16, how do autotrophs living in an aquatic ecosystem obtain carbon? Again, 3, 2, and 1. It's A. So, yes, it's dissolved carbon dioxide in the water and hydrogen carbonate ions. So they don't feed on heterotrophs, right? Because they're autotrophs. They don't eat other animals for their food. They also don't active transport carbon dioxide from the atmosphere because they're in the water, so the atmosphere is not available. And then they don't ingest organisms because, again, they're autotrophs, right? They do not consume other organisms. So, I mean, I think this is quite straightforward to realize it's A, right? But if you didn't get it, don't worry. If you have any questions, let me know below. So 17, in a natural classification, what do all members of a genus have in common? Again, 3, 2, and 1. D. Okay, A is false. They don't have the same name, right? I mean, by definition, there's different members in the genus that are different species, so they'll have different names. So they don't belong to the same species. B is also false. And then, so a genus has a lot of species in it, or at least 2. And then C, they can freely interbreed to produce fertile offspring. Now, that's the definition of a species, not of a genus, right? Therefore, it's D. They all evolved from the same common ancestor. That's why they're in the same genus. Okay, so question 18. A central idea in the theory of evolution is that species may evolve gradually over time from a common ancestor. Which statements explain the pattern of evolution seen in the diagram? Okay, stop to think now. And in 3, 2, and 1. It's 1 and 3. Why? Okay, so beak shape shows genetic variability in the common ancestor. That's true, right? Within the species, there was certainly variability amongst different organisms, the same way there's a variability in humans' noses, right? Then 2, changes in the beak shape that occur during the lifetime of an individual bird are passed on to the next generation. Okay, the key here is that occur during the lifetime of an individual bird. Remember, things that happen during your lifetime are not heritable, right? What's heritable is what's in your genes. If you scar yourself because you got injured, that's not going to be passed on to your offspring, right? So 2 is false. And then 3, changes in beak shape are heritable and make each species better adapted to its environment. Now, that is true. So the changes in the beak shape, the ones that are genetically encoded, do get passed on. So it's 1 and 3. Okay, question 19. What can be deduced from this cladogram? Again, I think quite straightforward. 3, 2, and 1. So humans and chimpanzees are more closely related to each other than to gorillas and baboons. You can see that, right? They're here, whereas gorillas are further away and baboons even more so. Why are the others false? So baboons do not share a common ancestor with the gorillas. That's false. It's here, right? Here's the common ancestor between gorillas and humans and chimpanzees and baboons. Then the common ancestor for all 4 groups is the baboon. That's not true. The common ancestor, again, is here at this node, right? The baboon diverged from that. And then the splitting events which separated the 4 groups took place at equal time intervals. That's not true because remember, in a cladogram, time intervals are represented by the length of the branch, and these branches are all of different lengths, showing different time intervals. Question 20. What is a function of the pancreas? Okay, 3, 2, and 1. It's B. So it releases glucagon when blood glucose levels are low. It also releases insulin when blood glucose levels are high, but not when they're low, so C is false. A, it does not release thyroxine, that's just false. And then D, it also doesn't secrete an endopeptidase. It secretes glucagon and insulin again. Okay, question 21. What occurs during the transmission of an impulse at a synapse? 3, 2, and 1. All of them. So all of them are true, okay? I'm not going to go over them because this is just textbook definitions of what happens at a synapse. If you want to take a look at that, then make sure to review the topic. But basically, remember, acetylcholine is released, right, with the input of calcium, and then that binds to the next membrane, and then it's reabsorbed, okay? So it's the recycling of neurotransmitters. Okay, so question 22. The diagram shows an alveolus from a healthy human lung with type 1 and type 2 pneumocytes and phagocytes. What are the main functions of these cells? Again, something you just have to remember. 3, 2, and 1. Okay, so it's D. So remember, type 1 pneumocytes, as you can see here, line most of the alveolus. So these are just the ones in charge of allowing gas exchange to happen, so CO2 and O2. Then type 2 pneumocytes, you realize there's a lot less. These are the ones that produce surfactants so that the alveolus doesn't collapse on itself. And then phagocytes, same in the alveolus as in any other tissue of the body. Phagocytes just try and ingest pathogens, right? They're the ones that try and eat pathogens up to destroy them. Okay. Question 23. Changes in membrane potential during nerve transmission along an axon involve the movement of sodium and potassium ions across the axon membrane. What explains the membrane potential at a Y in the trace? So here. Okay, take some time to think. This might be a bit trickier, but 3, 2, and 1. It's A, okay? So remember, beforehand, so after the resting potential and when we reach the action potential, that is sodium channels, right? But then when we go down in membrane potential, that is potassium channels, right? Which open and allow them to move outside of the axon membrane, okay? So sodium channels opening here, so C is false, right? Because that's not Y. And then D is also false because the change in voltage is always because of channels opening or closing. And here Y, when it decreases, it's because of the potassium channels, okay? Great. Okay, so going back to biochemistry, right? The topic on molecules. Amylose, amylopectin, and glycogen are polysaccharides that are components of human nutrition. Which molecules are products of the digestion of these molecules? Okay, so basically what are they made out of, right? 3, 2, and 1. So they're all alpha glucose, right? This is kind of a trick question. I think, remember, the only one that's beta glucose, okay, is cellulose. So hopefully you remember that, but all of these three are made of alpha glucoses bound together, albeit in different ways, right? With branches, no branches, etc., but they're all alpha glucose. Okay. Question 25, what is the role of the sinoatrial node? Okay. Again, factual. So 3, 2, and 1. Okay, so remember, the sinoatrial node is kind of the pacemaker, right? So it stimulates the contraction of the walls in the atria, and then that spreads to the rest of the heart. So it doesn't produce epinephrine, right? The heart doesn't do that. It doesn't send signals from the ventricles to the atria, and it doesn't ensure that the heart rate does not respond to signals from the medulla. So the sinoatrial node, basically the only thing it does is it makes the pace of the heart, right? It stimulates some cells, and then that spreads to the rest of the heart. Okay. So still with the heart. So the diagram shows a section through a human heart with the chambers, valves, and main blood vessels. What are the functions of X and Y? Okay, remember left side, right side, right? Okay, 3, 2, and 1. Okay, so I think Y is the easiest one, right? So the atrioventricular valves shown here prevent backflow from the atrium into the when the ventricle contracts, right? So when blood has come all the way here to the ventricle and it contracts, you want it to go down the artery. You do not want it to go back into the atrium, right? So I think that's pretty straightforward. And then X, okay, so it's the right side of the heart, right? So it carries deoxygenated blood to the lungs, right? And then again, that returns, remember, carries oxygenated, then goes to the organs, then comes back deoxygenated, and then the cycle repeats. Okay. All right, so next question. What is the function of DNA polymerase 1 in DNA replication? Again, pretty factual question. 3, 2, and 1. Okay, so it removes RNA primers and replaces them with DNA. So it allows replication to begin, basically, right? It doesn't form the primers. It doesn't build the leading strand by adding DNA nucleotides. Remember, that's DNA polymerase 3. And it doesn't form Okazaki fragments. So it's removal of the RNA primers, okay? Again, review this topic if you're not feeling too confident with it. Okay, question 28. The sequence of bases on a short section of the antisense strand of a gene undergoing transcription is shown. What is the sequence of bases on the resulting mRNA? Okay, you have to remember what the antisense strand is basically for this. In 3, 2, and 1. It's C, so it's guac from 3 to 5, right? So if you do the transcription of this, C to G, right? A to U, because thymine doesn't exist in RNA, T to A, and G to C. Okay, so basically, as you can see, if you were to superpose it over this, right, 3 to 5, it's the exact copy. So it's basically copying the antisense strand, right? And it would be identical to the sense strand, right? With the change, obviously, of thymine to uracil. Okay. Which statement distinguishes between the roles of free and bound ribosomes? Okay, I think this is pretty straightforward. 3, 2, and 1. Okay, so the answer is that free ribosomes synthesize proteins for intracellular use, bound ribosomes for export. Okay, so C and D don't make any sense, right? Because all ribosomes synthesize proteins, and then B is flipped around. That's just not true. So basically what ribosomes do, the bound ones, they're bound to the ER, and they use the ER and the Golgi to make those proteins leave the cell. Whereas the free ribosomes, you just make the proteins there and leave them to be. Okay. Next question is the production of isoleucine from threonine is summarized in the diagram. This process involves several steps and is regulated by, and this is key, feedback inhibition. What occurs during isoleucine production? Okay, again, 3, 2, and 1. Okay, so isoleucine inhibits threonine deaminase activity. How? You can see it in the diagram, right? Isoleucine over here is binding to threonine deaminase and inhibiting threonine from binding to it. Whereas you can't really see isoleucine binding to any of these at any point in the diagram, right? So that's why it's threonine deaminase and not any of the others. 31, and we're almost there. So what feature is common to both the Krebs cycle and the Calvin cycle? Okay, think carefully because this one's a bit tricky, I think. Okay, 3, 2, and 1. C. So they're not both catabolism or anabolism, right? Why? Because the Calvin cycle is anabolic, so it's building up something bigger. The Krebs is catabolic. And in terms of producing large amounts of ATP, not true. The Krebs does, the Calvin does not. But they both involve reduction reactions along the pathway. So hopefully that makes sense. Just, again, you have to recall what they're for and what they do. Okay, so what applies to chemiosmosis? And again, 3, 2, and 1. It's A. Okay, so it takes place in both cristae in the mitochondria and thylakoid membranes in the chloroplast. So that's true. It does not involve the movement of water across a semi-permeable membrane. It actually doesn't involve the movement of water at all. It only takes place in the presence of sunlight and oxygen gas. Not true. And it uses ATP to pump hydrogen ions against the concentration gradient. Not true. So actually, it does the opposite. It uses hydrogen ions to make ATP. Okay. So, next question. Phloem transports organic molecules from the leaves to other parts of the plant. How are phloem sieve tubes adapted for this function? And again, in 3, 2, and 1. So it's 1 only. Why? So 1 is just true, right? They have porous end plates, allowing the cytoplasm to move freely between adjacent cells. 2 is false because they don't contain a nucleus, right? So that's false. And then 3, there's not a high osmotic pressure. That's in the xylem, but in the phloem, there's not a high osmotic pressure, so they don't need thickened walls for that. Okay. Question 34. How does auxin cause plants to respond to changes in the environment? Okay. In 3, 2, and 1. So it controls plant growth in the shoot apical meristem. That's the main shoot, right? So it does not prevent cell walls from elongating. It does not cause shoots to grow away from the light. Actually, it's the opposite. And it does not ensure that plants are unaffected by changes in the environment. That just doesn't make... It's basically for growth, right? And it causes growth in the main meristem, and thus allows the plant to move to one side or the other to go towards the sunlight. Okay. Question 35. So chrysanthemums are short-day flowering plants that are often grown in greenhouses and sold commercially. Which conditions would ensure that they produce flowers at all times of year? And the key here is short-day flowering. So 3, 2, and 1. So uninterrupted darkness, right? Remember, short-day flowering plants flower when days are short, as the name says, so when it's dark a lot of the time, and long day when there's a lot of light, right? So basically summer versus winter. So if you wanted to grow these commercially throughout the year, then you would want to give it long periods of darkness because they're short-day flowering plants. Okay. So then the next question is, research into eye color in humans shows a range from very light blue or gray to dark brown or black. In Europe, eye color shows a strong correlation with latitude, with lighter colors being more frequent in northern latitudes and darker eyes more frequent in the south. This question is so long. What do these facts suggest about the genetic basis for eye color in humans? Okay. Again, 3, 2, and 1. It's a polygenic trait controlled by multiple genes. Why? Okay. So A is false because it's not determined by a pair of alleles, right? Otherwise, you wouldn't be able to have so many combinations. C is false because it's not environmentally determined and it is controlled by genes, right? And then it is not independent of environmental factors either because, as we can see here, different areas have different eye colors, right? And it's not discrete entirely either. But it is polygenic because there's many different phenotypes, right? Many different eye colors. And it's controlled by multiple genes as well because you have a lot of variation, right, in eye color. It's not just brown or blue, but you can have dark brown, light brown, hazelnut brown, etc., right? Okay. So that's why it's not discrete variation entirely. Okay, and 37, what is the role of calcium ions in muscle contraction? Again, pretty factual. So 3, 2, and 1. Yes, so they unblock myosin binding sites on actin, and they basically allow actin to bind to myosin, right, and then do the swing whereby muscle contracts. Calcium does not release energy at all. They also don't prevent cross bridges from forming. Actually, the opposite, they allow them to form, and they also don't cause myosin heads to bend, right? Well, the only thing they do is they unblock the binding sites on actin. Okay. Question 38. So what is the response of the immune system to first-time exposure to a pathogen? Okay. I think this one's pretty straightforward. 3, 2, and 1. So antigens on the surface of the pathogen stimulate B lymphocytes to produce plasma cells that in turn produce antibodies. Yes, that takes a long time, but that's exactly what happens in the human body. So antibodies are not present if it's the first time exposure, right? That happens only if it's the second or third time. The immune system will only respond to an infection if memory cells are present. That's not true. Again, memory cells make the response stronger, but they will respond either way. And then antigens on the surface of the pathogen stimulate the production of monoclonal antibodies? No. So monoclonal antibodies are, again, a technique, so they're basically man-made, right? So the immune system does not make monoclonal antibodies. So it's B. Okay, and 39. Which statement describes the function of the Malpighian tubules in insects? Okay, 3, 2, and 1. They carry out osmoregulation and excrete nitrogenous waste, mainly as uric acid. Remember, this is in the same topic as the kidneys, right? So that's what they do. They basically regulate the water concentration in insects, and they excrete waste as well. So similar to the kidneys, right? So it's nothing about excess nutrients being excreted, and insects don't secrete ammonia, they secrete uric acid. It also has nothing to do with gas exchange. And also, insects don't excrete urine, right? It's, again, uric acid. You need to remember the differences, right? And finally, what is the function of human chorionic gonadotropin, HCG? In 3, 2, and 1. So it stimulates the corpus luteum to produce progesterone during early pregnancy. So later on in the pregnancy, that's taken over by the placenta, but until then, that's what HCG is needed for. So progesterone is produced by the corpus luteum, and then the placenta takes over. Okay, so that's the end of paper 1, the time zone 1. And if you have any questions, again, as I said, leave them in the comments, and I'll see you next week for the next paper, which we're going to do, which is time zone 2. Have a great week, everyone. Bye.